All right, so let's continue with hearing anatomy. Um, we're going to look at the outer ear first. There's three different portions of the ear that we're going to talk about: the outer ear, the middle ear, and the inner ear. The outer ear is from the pinna to the eardrum. The pinna is this that you see over here on the left. This is your pinna. So pinna, everybody's pinna is slightly different. It's like a snowflake or a fingerprint. And actually, I was going to tell you a story that's like way TMI about humans. But you know what? I'm going to do it anyway. The artist Salvador Dali also figured out that the anus is as identifiable as a fingerprint. It's unique between people. So a snowflake and a fingerprint and that. And the pinna are all different from person to person. The pinna is usually what we think of as our ear. And the outer ear is going to go from pinna to eardrum, which is where the eardrum is kind of on the cusp of outer and middle ear. The grooves in your pinna actually do function. They do stuff for you. They're not just there to look pretty and be different from everybody else's pinna. They actually filter sound. So what you can do if you want to test this out is you can get Play-Doh and you can fill in the grooves of your pinna. And then if you listen to sound will sound different to you. If you just walk outside, like mask up and go for a walk, and you will not hear things the way that you normally do. And this is because the pinna is helping filter the sound. And you've learned what that pinna sounds like. I mean, to you, you've always had a pinna that's roughly the same shape. It's slightly different depending on how old you are, but as an adult, you've had the same shaped pinna since you stopped growing. And so to you, it doesn't sound like anything. You're just used to it. Kind of like how you don't see your nose until you start looking for it. Like we're just used to it there. Also, you're welcome. Now you're all looking at your nose. You're not going to not be able to see it in your vision for a couple hours. But once you cover up the pinna, then sound is a little off. One thing that we do when we make these holophonic recordings, which there's going to be a holophonic recording that you can listen to in this lecture, well, not in this lecture, but in this module. It's amazing. It really sounds like, it's called the virtual barbershop, and you really will feel like someone is cutting your hair, is using clippers near your ears. And you know what? Since we can't actually go out and get haircuts anyway, this is a sort of good, I guess, stand-in for that. So what they do for that, since the pinna is individual, they actually take moldings of a whole bunch of different pinnas, and then they average them together. So they create like the average pinna. And this is actually pretty good. It's good enough. This average pinna, this idealized pinna. And it's good enough to actually make you feel like you really are sitting at a barber shop. We have a microphone like this at with a Samba group that I work with. We applied for a grant. Like, it's an $8,000 microphone, so we needed a grant to get this, and I was the one who wrote the grant, so that was cool. It looks like a head. It's shaped like a face, and then it has ears with like really lifelike pinna. The face isn't super lifelike. It's just good enough to create a head shadow and to have sound bounce off of a nose in the way it would. So the idea of this microphone is you put the head somewhere and you record sounds with it, and when you go back and you listen to it, it sounds like you're there. It sounds perfectly like you are sitting there because it accounts for the head sound shadow and phase differences between the ears. The pinna is right. The way that sound bounces off the jaw and the nose and everything is right. It's amazing. And so far, we've mostly used it for like promotional photo shoots for the research that we do because the university likes the way that it looks. Anyway, that's the pinna. The pinna is really cool. There's so much that work that still we can do with knowing about how the pinna helps us hear. And then we have the external auditory meatus, which is the part that you stick your pinky into when you scratch your ear. It's also called the external ear canal. This also subtly changes the sound, not nearly as much as the pinna, but there is a resonance to it. Any tube is going to have a resonant frequency, and the ear canal is no different. It's a tube. It's a short tube, but it is a tube. So there is a resonance frequency there. For adults, it's between 2,000 and 4,000 kilohertz. For babies, it's higher, and that's because they're smaller. So a smaller tube is going to have a higher resonant frequency. And then, like I said, that just, filters the sound that we get. There's subtle changes from the sound out here to the sound in here because there's some filtering that's going on. Okay, so now we're going to talk about the middle ear. This is going to go from the eardrum to the cochlea. The middle ear is really amazing because it can amplify sounds up to 17 times. So really, really, really quiet sound, like a mouse whispering. You can amplify that up to 17 times so you can actually, hear these things. Leaves rustling very quietly. I remember one time it was like last summer or last spring, it was cool enough that I could open my windows, and I heard this like little rustling at night. And I'm like, what is that? I can't figure out what it is. And it's one of those beetles, those like green metallic looking beetles had like gotten behind my clock and was just walking around. And I could actually hear its legs like as it was walking behind my clock. So I was like, oh, weird, and threw it outside. But also thought like how crazy that was that I could actually hear that. It's such a quiet sound, but it was so silent in my house. It was the middle of the night, and I was like walking around, and I'm like, what? There's a sound. And so this is the middle ear amplifying things up to 17 times. But we also know that sound is damaging to our ears. So the middle ear also can help us dampen sounds so that we don't damage our hearing. We'll talk more about how that happens soon, but not yet. The eardrum is also called the tympanic membrane. It's a complete seal against the outside to prevent you from getting infections in your middle ear. A middle ear infection is not great. An outer ear infection is also not fun, but it's not usually something that's going to cause a whole lot of damage. Middle ear infections can cause damage if they're not treated right away. But outer ear infections are usually fine. They're easy to treat. You just put some like Debrox drops in, and it takes care of the infection, and you're okay. Swimmer's ear is one of the outer ear infections. Middle ear infections are definitely a whole different problem. In fact, if they're really bad, they can cause hearing loss. Really bad middle ear infections. There's a lot of middle ear. I don't want to freak you guys out. I mean, middle ear infections usually don't result in anything beyond I don't feel very good, in my ear, and maybe there's some pus or something that comes out, and then you get medication, you're okay, and you never lose your hearing. But they can progress to that stage. So the eardrum the eardrum is this seal that protects against infections, or it tries to. Sometimes they still happen. There's also three important bones inside the ear, and these working together create the leverage that allows the sounds to be amplified the 17 times. That's the malleus, incus, and stapes, or the hammer, anvil, and stirrup, as you might know them. And here's a good picture of what. So here, this is the pinna. This is the external auditory meatus right here, going through the bone of the skull right here. And then here's the eardrum. And then this is the ossicles right here, those three bones, which is called the ossicles. This is a close-up. So you've got your hammer, which directly interfaces with the eardrum. You've got the anvil, and then the stapes or stirrup. And this is going to interface directly with the oval window of the cochlea, which is where we're going next. That's the inner ear. And we'll talk a little bit more about muscles and stuff that go along with this in a few seconds. All right, so this is the middle. Oh, we're still talking about the middle ear. Oh, this is when we're going to talk about the muscles. I thought I had a different slide later on about this. So you have these important muscles that interface with the ossicles right here. The tensor tympani and the stapedius muscle. They're both going to innervate and tighten up, and they prevent the tympanic membrane from moving as much. So if the tympanic membrane doesn't move as much, that means that sound isn't transferred through the ossicles, which means that not as much sound enters the cochlea, which means the hair cells aren't pushed around as much. And the hair cells are the things that actually die when you listen to sounds that are too loud. It's actual cell death, and you can't regenerate it. We can't regenerate it yet, I should say. And it could be a long time before we can. So this helps protect us against that. If you've ever been to a really loud concert and you kind of felt like a tingliness in your ears, or like they were shivering, this is the tensor tympani and the stapedius muscle. If you're a person who can ear rumble by just tensing up, yeah, there we go. I'm a rumbler. Anybody else a rumbler? Let me know. But if you can make your ears rumble, that's what's going on. You're tensing up your tensor tympani and stapedius muscles and just tightening up that eardrum. You can actually, if you can do that at will, you can actually do that before there is a loud sound and help protect your ears. So let's say the 4th of July is coming up, somebody has fireworks. If you put your fingers in your ears or cover your ears and tense up your these muscles that hold the ossicles in place, then you're doing a good job to protect your hearing. Okay, we'll move on. So we're going to talk about the cochlea. Well, let me go back over this for 1 second. So remember, the stapes right here is going to interface with the oval window. What it's going to do is it's kind of like, think about a bass drum, and the bass drum has this thing that like just kind of like keeps pumping against the drum, right? So you've got this, I don't know, it's like a hammer. I don't know what it's called. I'm not a drummer. But it thumps against the actual drum. And that's kind of what the stapes does against the oval window. Except that instead of a drum head, it's actually this kind of opening. So it's going to keep hitting this. Now what that does is it creates a wave. So this is when we're going from a sound pressure wave. The sound pressure wave comes into the ear, and then it hits the eardrum. Trying to think of the actual word. I just said it, and now it's left my brain. Whatever, it doesn't matter. The tympanic membrane. So sound pressure waves come in, they hit the tympanic membrane. The tympanic membrane deflects, and it moves the ossicles. Now, the wave has become a physical movement. It's no longer a sound pressure wave. It's a physical movement. Here. But then, the physical movement is hitting against the oval window of the cochlea. The cochlea is a fluid-filled structure. And this part hitting against this open oval window of the cochlea is now creating a fluid wave, like a wave in the ocean, right? So we've gone from a sound pressure wave to mechanical movement, rhythmic mechanical movement, to a fluid wave in the cochlea. So we're transducing in all these steps. Sound pressure wave, rhythmic mechanical wave, and then a fluid wave. So the cochlea is this fluid-filled organ. It's spiral-shaped. It's not actually, I mean, we say it's spiral-shaped, but that gives you the idea, you know, that it's shaped like this, like a perfect spiral. It's actually more like a poop emoji, like it, or like a snail. Yeah, poop emoji is pretty much the best. It kind of comes up like a poop emoji. And so that, I think, is the best way, really, to conceptualize it. We always talk about it being spiral, but it's not a perfect spiral. It's a poop emoji. And then you can see that if it is shaped like a poop emoji, this inner part down here is always going to be facing in on itself, right? Because it's kind of spiraling up, kind of like a helical structure and spiraling. But the bottom portion of the cochlea is always going to be facing in. I guess I should say the inside portion of the cochlea is always going to be facing in. The outside portion is always facing out. Whereas if it was a true spiral, you'd have the inside portion touching the outside portion, and that it doesn't work as well. Because the inside portion, this down underneath part in this view down here, this is where the actual like nerve is that's going to send signals to the brain, which we'll get to in a second. So anyway, we create a wave. The wave has its maximal amplitude at the position on the basilar membrane that corresponds to the frequency of the wave. So again, we're talking about cycles per second. So if the stapes is hitting against the oval window 4 times a second, it'd be 4 hertz, which we can't actually hear. I'll come up with a different one. 400 times a second. So that's going to be 400 hertz. Then the fluid wave is actually going to deflect the basilar membrane, which is this membranous portion of right here in the center. You've got this basilar membrane. So it's going to deflect maximally at a place corresponding to 400 hertz. It sets up this standing wave right there, and it just keeps being deflected at 400 hertz. And what's cool about that is we can tell that the cochlea is tonotopically arranged. 400 hertz is here. That means 300 hertz is over here. 200 hertz is over here. 600 hertz is further up. 1200 hertz is even further down. Tonotopically just means arranged according to frequency or pitch. It's actually frequency, because pitch is determined in the brain. And the cochlea, we're not quite at brain status yet. This is where we're about to interface with the brain. So it would be frequency. And the tonotopic arrangement of the cochlea looks like this picture right here. So here's by the oval window. 20,000 hertz is right here. Now you should remember from earlier in the class, we were talking about the human range of hearing. It goes from 20 hertz to 20,000 hertz. 20 is very low. 20,000 is very high. I mean, it's unlikely that anybody actually hears at either of these extremes, but this is the range, the possible range of human hearing. 20 hertz to 20,000 hertz. And right here, by 20,000 hertz, this is where the stapes and the oval window are. And what that means is, it doesn't matter what frequency you're hearing. It can be high, it can be low, it can be very low, it can be very high. It's passing over 20,000 hertz. There's going to be a wave that passes over 20,000 hertz, and that's going to cause the hair cells to move near 20,000 hertz. If it's a high sound, like let's say it's 7,000 hertz, it's going to deflect the basilar membrane only down to here, only down to 7,000 hertz. The rest of it doesn't move at all. If it's a 200 hertz tone, it's going to cause all of this entire organ, the basilar membrane across this entire organ, to move. But what's important about that is this portion up here always moves. Always. For any sound. Where do you usually see hearing loss? High frequency. And why is that? Well, because all sounds pass over that portion of the cochlea. All sounds pass over this high frequency part right here. And over time, the hair cells in that region start to get worn out. The cells start to die. They get damaged. And so that's why we have high frequency hearing loss. And this is common among people, just it's a common byproduct of aging. Sure, some people will have it more because they've been to a lot of rock concerts or they work around a lot of construction things. But just the fact that you are a living human who hears means that you're going to have high frequency hearing loss. Okay, so let's look at more parts of the cochlea. So this slide isn't super important. This is maybe a little more specific than it needs to be. You'll get into this when you take hearing science. But what's important about this is here's the tectorial membrane. Here's the basilar membrane down here. When the basilar membrane deflects and moves up, it's going to push against the tectorial membrane. Here's the hair cells right here. So the tectorial membrane is here. The basilar membrane is here. The tectorial membrane is here. The basilar membrane is going to deflect up, which means then there's hair cells here, which are going to get pushed against the tectorial membrane. The act of pushing is what releases the neurotransmitter that then causes the nerve, the cranial nerve, the eighth cranial nerve, or the auditory nerve, to fire. So unlike a normal neuron, the hair cells don't actually fire by getting impulses from other nerves because they're the first nerve. There's nothing that can signify them to fire, that can signal them to fire. So what has to happen is there's something mechanical, and that's the basilar membrane pushing against the tectorial membrane, which then pulls on these hair cells, just like this. And that releases neurotransmitters, which then causes other neurons to fire. Let's take a closer look at the hair cells here. So again, this is what I said. Pushing the end of the hair cell allows for an electrical charge to escape, which is actually not 100. I mean, it is true. It's a depolarized ion that's escaping. It's an electrical charge. Whatever. You don't really need to know the exact, at least not at this stage of your career. When you get into your master's program, you'll have to know the differentiation, but essentially it's an electrical charge. Pushing on these hair cells creates an electrical charge, which then depolarizes the neurons further down the chain. You have two kinds of hair cells, outer hair cells and inner hair cells. The outer hair cells, which you have more of, are kind of like a preamplification system. They amplify low intensity sounds, which we already know that the ossicles can amplify quiet sounds. But what's going on here is something more akin to compression. So the hair cells will amplify quiet sounds, they'll dampen loud sounds. And what compression does, this is like an audio person thing, like if you're into audio, you might know about compression, but it takes this dynamic range. So like you have loud sounds that are way up here, you have quiet sounds that are way down here. And what you do is you take the really loud sounds and you limit them. You take the quiet sounds and you boost them. So you compress your dynamic range. And that's essentially what these 3 rows of hair cells are doing too. They're compressing the range of what you're actually listening to. One of the things that we encounter with microphones is like this microphone that I have here. This is a great microphone, but it doesn't have dynamic compression. And so there are things that I can do, normal ways that I can talk. I can like get really quiet, or I could totally just shout. And I'm watching my levels over here, and I see that I've peaked when I shouted, and when I was talking really quiet, I think there were things that it wasn't catching. And the other thing is I can hear things happening in the room that I'm recording in, like papers are moving because of my fan. I can hear them. They're not even showing up on the microphone because this dynamic range is too large and it doesn't have dynamic compression. So what we're doing is we're hearing all of this at the same time, and our outer hair cells are making it so, yeah, we still hear that it's louder or quieter, but we can hear it all. It's not like we are just tuned into this really loud sound and we can't hear the quiet sounds, which is what your microphone is. You can turn down the gain, and then loud sounds you can hear, but all the quiet stuff you miss. Or you can turn up your gain, and then you hear quiet sounds, but the loud sounds just blow out all of your speakers. So anyway, that's what the outer hair cells do. The inner hair cells are the ones that then cause the firing of the next neurons. So the outer hair cells are here, they're doing this dynamic compression, then the inner hair cells are actually firing. They're being pushed on, firing, and making neurons further down the chain depolarize and fire. Let's see, we should go into, we're going to go into neuroanatomy in a second. So our hair cells can't regenerate on their own. Once they're dead, they're dead. And they're damaged by exposure to loud sounds. Or like we just said, they're also damaged just by constant exposure to sound. So you can be exposed to a loud sound for a short amount of time and have damage to your hearing. Or you can be exposed to a slightly loud sound for a long period of time and have damage to your hearing as well. I know a dude who used to fly fighter jets back in the 70s, and that was a constant high-pitched whine that if you were in it for a couple minutes, you probably would be fine because it's not super loud inside the cockpit. But the fact that this guy had hundreds, possibly thousands of hours of, definitely thousands of hours of flight time, probably 10,000 hours of flight time or more, he had to wear hearing aids that boosted his hearing quite a bit because of that constant exposure to that loud whining noise. So we can't regenerate our hearing yet. There are some studies that are somewhat promising that show that we can encourage cell growth in guinea pigs and chinchillas, but we can't do it in humans yet. So they're trying to figure out how we can do that. They're trying to figure out that there's some stem cell research that we can get our hair cells to regrow, so that instead of wearing a hearing aid when we all get old, that we can just pump stem cells into our ears and then hear again, which would be great. But it's probably pretty far off. And as of now, once the hair cells are gone, they're gone. So this is time for a PSA for me to say, don't listen to your music super loud. Like I used to go, when it's not quarantine, I really like to go to cool concerts. And the last time I went, I did not bring earplugs. I should have. Like it was still really fun, but then the next day you're kind of like, feel like you got cotton in your ears. There's definitely some hearing loss there. And when I was in high school, I went to concerts all the time. And it'd be like a day or two that my ears would feel funny afterwards. And I know that that's hearing loss. So, I mean, you can still go, but put hearing aids in. And don't use these kind of headphones. These are actually not super great. Earbuds are good because they can block outside noise, so then you don't have to turn your music up as loud to drown out everything else. Really good over-the-ear headphones are also good because they also drown out outside noises. These are kind of like the crappy in-between zone where they're not around your ear enough to drown out outside noises, so you have to like boost the volume way up so you can actually hear what's going on with your music or your podcast or whatever. So these are not the greatest kind of headphones. But anyway, take care of your hearing. Always wear hearing protection when you work with loud machinery, because once it's gone, it's gone. Okay, so let's talk about neuroanatomy of hearing. So once we get to the hair cell, once we get to the inner hair cell, it interfaces with neurons in the 8th cranial nerve. The 8th cranial nerve is also called the auditory nerve. And then from there, that's going to go into the cochlear nucleus, which you can see right here. So here's our auditory nerve coming up from the hair cell, and then boom, cochlear nucleus. Now from the cochlear nucleus, it goes to a bunch of little different complexes up through the brainstem, the olivary complexes, the different areas. It's not something that you need to know right away. But one thing that is good to know is that about 75% of that information crosses over to the contralateral side. Contralateral means opposite. So if it came from your right ear, it goes over to the left side of your brain. The contrary to that is ipsilateral, which means it stays on the same side. So the other 25% of auditory information comes into the right ear, and it remains ipsilateral by traveling to the right side of your brain, the right auditory complex. Not complex, the right auditory cortex, not complex. It's probable that the reason why 25% of this stays on the ipsilateral side is for localization and other things, though we're not 100% sure. But we do know that we have something called the right ear advantage for speech. Why do you think that might be? What side governs speech? Do you remember? In most people, it's usually the left side. So we know that 75% of auditory information crosses over, so the right ear is somehow better at listening to speech than the left ear. The right ear advantage means that you're usually able to be more accurate and also faster at responding to words that are given to your right ear than you are to your left ear. And again, that's probably because most of your auditory information crosses over and goes to the left side of your brain, which is where speech is governed. Another word for the primary auditory cortex, or another term for the primary auditory cortex, is Heschel's gyrus. And that is also tonotopically organized. Just like, what else was tonotopically organized? Yes, the cochlea. So what you can see here, this is pulling back part of the brain and looking at the primary auditory cortex right here on the temporal lobe, the top portion of the temporal lobe, is that you see that 500 hertz is down here, 1,000 hertz is slightly above that, and then 16,000 hertz is all the way at the top. So that shows tonotopic organization of Heschel's gyrus. So you would expect that if an area on the cochlea that corresponds to 500 hertz is active, the area in Heschel's gyrus that corresponds to 500 hertz would also be active. And so then the areas around the primary auditory cortex and the Heschel's gyrus, those are involved in the strictly auditory perception. So there's a lot that goes on in the temporal lobe, but the part of the temporal lobe right around the primary auditory cortex is dedicated just to auditory processing. And the further away from the primary auditory cortex it gets, the more high level it becomes. So right at primary auditory cortex, it's kind of like just sensing individual pitches, and then as you move out from there, it turns into complex tones and words and music and things like that. And that is the end of this lecture. We're going to talk about hearing disorders next. Yeah, I definitely hit the wrong. Sorry, you're just going to have to. Okay, there we go.